Hello, I'm Luke O'Neill and here I am in my lab in the Trinity Biomedical Sciences Institute. So this week on my COVID-19 update, blood clots. Clotting is yet again still in the news, and as we know, the AstraZeneca vaccine had an issue, very rare clotting events in the brain and abdomen, and Johnson Johnson, very similar, and the EMA have said it's the same disease really caused by both vaccines. What's very interesting is they've got a treatment now to treat this clotting disorder. It's a thing called IVIG, and we had the first report of a woman presenting with this very rare clotting disorder. She had a rash, actually, it was one of the main symptoms. They gave her IVIG, and guess what? The clots went away and she was discharged very quickly. And the authors conclude a rapid response will now stop this clotting disorder with the AstraZeneca vaccine. Now what this means is it gives authorities more wiggle room now. They may well approve it again for younger people because now this rare clotting event can be treated. Every hospital has IVIG in the fridge, so it's very easy to treat. So therefore we may see AstraZeneca and Johnson Johnson being more widely used again. Interesting stuff as well on this very rare type of clotting, which is called VITT. Guess what? The natural infection has a risk of it. It's a 1 in 28,000 chance that the normal infection with COVID-19 will give you this very rare clotting. And of course, guess what? The vaccine is 1 in 150,000. So you're at a much higher risk then of actually getting these rare clots by being infected. So yet again, that suggests we shouldn't really worry about the vaccine causing these clots. And a study in Stanford, they've taken the Pfizer and Moderna vaccines in the lab, analysed them, great details on what they're made of, and released the information into a thing called GitHub. Now, isn't that striking? Now, GitHub is a place where you publish software usually. And what they've done is they've given the sequence of the 2 RNA vaccines, and anybody now can read the sequence of both of those vaccines and make it in the lab. I mean, it's very interesting what this means for intellectual property. The Pfizer vaccine sequence was out already in the public domain, and now the Moderna vaccine sequence is available. This could be useful for developing diagnostics, say, to detect the virus, or maybe even developing vaccines anyway. And of course, the big question is now, will some people use that information now to develop their own vaccines? So a very interesting development. And an interesting development as well is about future vaccines. Obviously we have the vaccines at the moment and they're great, but there are 22 new vaccines in development that will stop any COVID-19 virus, any variant of it, and maybe even coronaviruses yet to jump from, say, bats into humans should that happen. These are called pan-coronavirus vaccines, and they're being designed to recognize a whole range of different coronaviruses. And a second study has shown you can take the spike protein now, which is what the vaccine is often against, or made against, and you can stick the spike protein onto these nano cages, tiny little structures, stud them with the spike, you're kind of making a viral-like particle, and use that as a vaccine. And they're using all these different types of spike proteins from all the variants. So again, you never know. Quite quickly we may have vaccines that will work against any variant of COVID-19. It'll take time, of course, to develop these and for them to be tested. We're looking towards the autumn, I guess some of these vaccines may be available. But again, the variant issue then becomes less of a worry because these vaccines are in development. And you can hear about these stories and more on my weekly COVID-19 update with Pat Kenny on Newstalk.